Hello everyone, this is LATHA AKULA. In this video, we are going to study about the mechanism of enzyme action. So there are two hypotheses proposed for explaining the enzymatic action. They were lock and key hypothesis and induced fit hypothesis. Lock and key hypothesis. So here, if you see this lock and key hypothesis was proposed by Emil Fischer. Who proposed this? Emil Fischer. He proposed lock and key hypothesis. According to him, both enzyme and substrate have the specific geometrical shapes. What they have? They have the specific geometrical shapes are present which are similar to the lock and key. If you see, the specific key only able to open the specific lock. Not all the keys will open the locks. Like this, enzymes also, they were specific to each other. Active sites have special groups. So in the enzymes there are active sites, right? So in that active sites there are some special groups like NH2, COOH, SH groups are present. So these groups they're helpful in establishing the contact with the substrate molecule. Just like the lock, how it can be opened by the specific key, the substrate molecule can also be acted upon by a particular type of an enzyme. In this, what we have studied, enzyme binds with the substrate. The substrate will binds to the active site of a particular enzyme, and after binding it forms the enzyme substrate complex. Within the enzyme there are several special groups are present, right? So because of the presence of those special groups, the substrate establishes contact with it, and that substrate is converted into product and form the enzyme product complex. After the formation of enzyme product complex, the enzyme and product, both of them, they get dissociated and product will be released into the surroundings, and enzyme will be binds to another molecule of the substrate. This is called as lock and key hypothesis. So what is the second one? Induced fit hypothesis. So this induced fit hypothesis was proposed by Koshland. Who proposed this? Koshland in the year 1960 he proposed. Here in this model, initially the active site of the enzyme that does not exist in the shape of the complementary with the substrate. But whenever this substrate comes and bind with the enzyme, it gets induced to assume the complementary shape as the substrate becomes bound to the enzyme. So here what is happening? The active site of the enzyme is very flexible here. Whenever the substrate binds to the active site of the enzyme get induced to assume the complementary shape substrate and fit around the substrate. Hence it is called as induced fit hypothesis. Next one is inhibition of enzyme activity. So up to now we have studied about different factors that affect the enzymatic activity and mechanism of enzyme action. Now we will see how the enzymatic activity can be inhibited. Here, what is inhibition or inhibitor? Any substance that can diminishes or damages the rate of speed of reaction or the velocity of the enzymatic catalyzed reaction, that is called as inhibition. There are two types of inhibitors are present. One is a reversible inhibitor and second one is the irreversible inhibitor. If you see the reversible inhibitor, this reversible inhibitor that binds with the enzyme's active site non-covalently. That means loosely. Non-covalent bond is a loose bond, right? Not strong as as like the covalent bond. So the non-covalent bond, and we can separate this enzyme and a reversible inhibitor by diluting this. And whenever this enzyme and inhibitor complex was diluted, both of them get dissociated into enzyme and a reversible inhibitor. So next one is irreversible inhibitor. So in irreversible inhibitor, enzymes and irreversible inhibitors, they bind covalently. That means there forms covalent bonds, and that is very strong bonds, right? That tightly binds to the enzymes and which cannot be dissociated even if we have diluted the enzyme inhibitor complex. Examples for this irreversible inhibitor was insecticide. Insecticides means which kills the insects, right? Some insecticides, they act as neurotoxins. Neurotoxins means they damage the nervous system and kills the insect. That type of insecticides are called as neurotoxic. Example insecticide is acetylcholine esterase. Acetylcholine esterase is one of the enzyme. And if we see these two are the inhibitors. Now we will see what are the types of inhibitions. Types of inhibitions. There are two types of inhibitions. One is a competitive inhibition and second one is a non-competitive inhibition. Competitive inhibition means there is a competition between the enzyme and inhibitor. For example, for instance, see here, this is the enzyme, this is a substrate and inhibitor. See here, both of them similar to each other, right? So both of them want to bind with the active site of enzyme. There is a competition between the inhibitor and substrate. Between these two compete with each other, hence it is called as competitive inhibition. And here see, instead of substrate, inhibitor binds in the active site of the enzyme. Finally what will happen? The substrate will not binds with that and there is no action. Enzymatic activity takes place. There is no product formation occurs. So this type of reaction is called as competitive inhibition. Now we see what is the effect of competitive inhibitor on Vmax. The effect of competitive inhibitor, it is reversed by increasing the substrate concentration. Here the substrate, it has to dominate the inhibitor's competition, then only the reaction will proceed. If we have not added any substrate, if we have not added extra substrate, the reaction will be stopped. To prevent that, in the presence of competitive inhibitor, more and more amount of the substrate is added. The substrate concentration that dominates the inhibitor's concentration, then the more substrates will bind to the enzyme active sites and which are not available for the inhibitors. So in that condition, at sufficiently high substrate concentration, the reaction velocity that reaches to the Vmax, but the reaction is slow. The reaction will be slowed down, and compared to the reaction without inhibitor, the reaction with inhibitor, it is a very slow reaction. So see here, there are two graphs are present. So in one graph, the reaction is normal without any inhibitors. In that condition, happily the enzyme and the substrate will binds to the active site of an enzyme and the reaction will be proceeded, and at particular time that reaches to the Vmax. That is okay. But the reaction with competitive inhibitor, it has to slowly and very hardly, it reaches to the Vmax very slowly. So the substrate has to compete with the inhibitor, at the same time the reaction need to be proceeded. So in that condition, of course it reaches to the Vmax, but it takes more time when compared to the normal reaction. So in the Vmax, what is the effect of competitive inhibitor on Vmax? The reaction will be slow. The reaction will be slowed down. Here, half Vmax. Now we'll see what is the effect of competitive inhibitor on Km. So in the presence of competitive inhibitor, the Km value increases. See here, this is a normal Km and this is the Km with the inhibitor. So the Km value will go on increased because we are adding the more and more amount of the substrate, right? That is why the Km value also increases. So in the presence of competitive inhibitor, more substrate is needed to achieve the half Vmax. See here. So when it has to reach to the half Vmax, more substrate is added and then only reaction will reach to the half maximum. So in the presence of competitive inhibitor, Km value increases, whereas the Vmax value, the time will take more and more amount of the time is taken by the Vmax and the Km value increases. That is the effect of competitive inhibitor on the Vmax and Km. Now we will see what are the examples of the competitive inhibitors. Now we will see the examples of the competitive inhibitors. In that the main examples are the sulfur drugs, next ethanol and melanate are the main examples of the competitive inhibitors. Now one by one we'll see about them. The first one is Ethanol, how the ethanol will act as a competitive inhibitor. So this ethanol will act as a competitive inhibitor for the methanol. So where it acts, this ethanol binds to an enzyme alcohol dehydrogenase. Actually, here the methanol has to bind here. For methanol, this ethanol will binds to the alcohol dehydrogenase and prevent the formation of formaldehyde. The production of the formaldehyde will be slowed down here. The next example, antibiotics which are used to kill the bacterial pathogens in the human beings. So here happen is, normally for any cell, if it has to grow, it needs folic acid. But bacteria, the folic acid from the cells will not be reached inside the bacterial cell wall by means of any active transport or diffusion. So the bacteria must have to synthesize its own folic acid. For that, it needs para-aminobenzoic acid, which is in shortcut it is called as PABA, para-aminobenzoic acid. Here, these sulfur drugs, they mimic the para-aminobenzoic acid. See the structures of para-aminobenzoic acids and the sulfanilamide, both are similar. Here this sulfur drug will mimic the para-aminobenzoic acid and act as a competitive inhibitor. So because of these sulfur drugs, the folic acid synthesis will be stopped and the bacterial growth will also be stopped in the body. Then that leads to the decrease in the number of bacteria, which lead to the control of the disease. Like this, the sulfur drugs will control the diseases, bacterial infections. The next one is melanate. Melanate acts as a competitive inhibitor. It will binds to the enzyme succinic dehydrogenase. Which enzyme? It binds to the succinic dehydrogenase enzyme, where it competes with the succinic acid. Non-competitive inhibition. So here, like inhibitor, this non-competitive inhibitor will not binds to the active site of an enzyme. Here, when inhibitor and substrate bind at different sites on the enzyme. The non-competitive inhibitor can bind either free enzyme or bind to the enzyme substrate complex, and finally the reaction will not be proceeded properly. It prevents the reaction from occurring. So what is a non-competitive inhibition? Here the inhibitor and substrate binds at different sites. Substrate binds to the active site and the inhibitor will binds to the some other place instead of the active site of the enzyme. So here it will not compete with the substrate, but instead of that, this non-competitive inhibitor will bind to either free enzyme or to the enzyme substrate complex and the reaction from occurring. And now we'll see what is the effect of this non-competitive inhibitor on Vmax. So in the competitive inhibitor, to overcome the problem of this inhibitor, what we have done, we have increased concentration of substrate, but it cannot be overcome by increasing the concentration of substrate here. So we cannot overcome the problem of non-competitive inhibition in this by increasing the substrate concentration. So automatically what will happen? The Vmax of the reaction decreases. See this graph. Here, this is the curve without inhibitor and this is the curve with non-competitive inhibitor, the reaction with non-competitive inhibitor. Slowly the reaction was decreasing, right? So by increasing or decreasing the substrate concentration, we cannot overcome the problem of the non-competitive inhibition, and then finally what will happen? The reaction decreases. So what is the effect of non-competitive inhibitor on the Vmax? The Vmax of the reaction decreases here. Next, effect of non-competitive inhibitor on Km. The non-competitive inhibitor do not interfere with the active site of the enzyme, right? So there is no problem for the substrate for binding site. The enzyme shows same Km in the presence or in the absence of the non-competitive inhibitor. The same Km will be seen in both the cases. So there is no change in the Km value. See the presence of inhibitor and in the absence of the inhibitor, same type of Km is seen. There is no change will be observed. Whereas in the competitive inhibitor, the Km value increases because there we have increased the substrate concentration. But here same type of Km be seen in both the cases. So what is the effect of non-competitive inhibitor on Km? The non-competitive inhibitor do not interfere with the substrate binding site of an enzyme. That shows the Km in the presence or absence here, the same type of value is same. What is the example for the non-competitive inhibitor? The cyanide is the poisonous chemical substance that kills the animal by inhibiting the cytochrome oxidase enzyme. What is the cytochrome oxidase enzyme? This is the enzyme involved in the electron transport chain. So by inhibiting the cytochrome oxidase enzyme, that kills the animal. So this is the non-competitive inhibitor.